Yes, this is Mansur, Government Degree College, Kamarati, lecturer in Zoology, Department of Zoology and Fisheries. Today topic is Cycon skeleton. Today topic is Cycon skeleton. The skeleton which helps to protection of the body. See, a skeleton. The skeleton composed of tiny pointed structures made of silica or the calcium carbonate called spicules, which are the embedded in mesenchyme. The second point is tell us skeleton may also be made of a network of protein fibers called spongin, and these endoskeleton protects and supports the softer parts of the body and calcareous spicules or large spicules. See one's structure. This is a skeleton of the cycon. Here the skeleton composed of tiny pointed structures, like in a tiny pointed, this is calcareous. This is calcareous tiny pointed structures completely, and these structures is made up of silica or the calcium carbonate. So that's why this called as the spicules, which are the embedded in the mesenchyme. It is a production from mesenchyme. Now, second point is that this is the skeleton may also made of network of protein fibers called the spongin. This spongin, Spongylidae, they have completely skeleton made up of a spongin. Like all their skeleton, all these like spicules is by protein fibers. And these endoskeleton protects and supports and protects and supports the softer parts of the body. So these only skeleton which helps to just only protection of the body. Here the calcareous spicules are called large spicules. Here this all these calcareous spicules is the large spicules, and the spongin spicules, so this is very protein fibers, and the siliceous spicules and demisponges is clear. See that, and siliceous spicules is exactinelida very clearly. Okay, this is the skeleton of Spongylidae. See the next very clearly, and next types and structure of the spicules. Here the different types of structures is there and types of spicules. See and triaxins and tetraxins, exactin and monaxin and tetraxin and prolactin. In different types of spicules is there in poriferans. See one structures very clearly. Very clear, the spicules and types: triaxins and triactin, exactin, monaxin, and tetractin and prolactin. All these structures. Okay, then second development of spicules, and where it is the developmented very clearly. Here the secreted by the specialized amoebocytes known as the scleroblasts. The secreted by the specialized amoebocytes, and the spicules is development in the special parts is that this is the amoebocytes, cells is that this is amoebocytes known as the scleroblasts. This scleroblast which helps to just only projection of the spicules. And a monaxin spicule is formed by the two sclerocytes and produced by the division of one scleroblast. And a triaxine and spicule is formed by the 6 sclerocytes produced by the division of 3 scleroblasts. What are they? See once the structure. This is development of spicules. See once founder cells and thickener cell and sclerocytes and the scleroblasts. Here the and secreted by the specialized amoebocytes known as scleroblast. And second point is the monaxin spicule is formed by 2 sclerocytes produced by the division of one scleroblast. And third point is that this is the triaxin spicules, like triaxin, which means of 3 spicules, 3 shapes is here, formed by the 6 sclerocytes produced by the division of 3 scleroblasts. Clear. And see once the water circulation, how to get water circulation in the poriferans. Clear the from weather conditions to body and body to weather conditions. Clear. See, the water current used in feeding, gas exchange, removal of wastage, and release of the gametes. Here, the water circulation, which helps to 3 functions, is there. This is the first function is the feeding, and the poriferans, the organisms depend on the feeding, depend on the feed, nutritions. Like and they live to because only feeding system. The current used in the feeding. And second one is the gas exchange. Second one is the gas exchange. That's why the gas exchange, which means of the CO2 carbon dioxide. CO2 carbon dioxide. And how to get CO2 and how to release to CO2 and how to get oxygen. And the removal of waste and removal of waste, which means of the food materials and the wastes and excretory products, and the release of the gametes. Here the gametes, which means of the release gametes, are the worms and the sperms. Then because of the sponges are the sessile, and all these animals, sessile animals, and these animals attached to the substratum. And the third point is that water current is cast by the constant beating of the flagella of choanocytes. Here the water current is cast by the constant beating of flagella of choanocytes. And the next fourth point is brings food and oxygen and removes the excretory and reproductive elements. And brings food and oxygen and removes and excretory and reproductive elements. See the course of water current, and the first is these animals where it can live. It can live in the weather conditions like water areas. Here the outside water, how to get, and the first is they have completely dermal ostia, and dermal ostia is always absorption to the water and expose the water. Here the water will enter into the dermal ostia, and dermal ostia connected to the in-current canal, and in-current canal is attached to the prosophile, and the prosophile is between the radial canal and the in-current canal, and this prosophile which elaborated and elaborated at the same time is they are the same sizes will be increased and decreased. Then the radial canal is through the epiphyll and the excurrent canal, and excurrent canal is and opens to the spongiocele, and the spongiocele was in the asculum. Asculum is pushing the outside. This is the water current. This is the course of water current. Once again, see clearly. The structure is very clear. The water circulation. See, look once. This is the water circulation. The water current is in feeding, gas exchange, removal of wastes, and release of the gametes, and because of the sponges or sessile, and the water current is cast by the constant beating of flagella of choanocytes, and brings and oxygen and removes excretory and the reproductive elements. And the course of water current, here the outside water like ostia, in-current pore and the spongiocele and asculum outside. Here the course of water current. They very clear the course of water current. Shall we discuss just a few minutes before? Outside water and the dermal ostia, enter the dermal ostia and the in-current canal, and the prosophile, radial canal, and epiphyll, and the excurrent canal, spongiocele, asculum, and then after other outside. This is